Vaccination is already started in US?
Oh yeah, we have vaccination is in our supermarkets now. So
Okay.
Wow. To start it, we have around 50 students who will be listening to you.
okay.
From different backgrounds of, well, this is the Biotechnology Institute. I can brief you about the institute. So this is the
Yep.
regional center for biotechnology, which is started by Government of India, Department of Biotechnology, which comes under Ministry of Science and Technology. So it starts with the name regional, keeping in the sense that we need to train the students in research, both for masters as well as PhD from the SARC regions. It comes and it is affiliated by the UNESCO. So with that, the regional name came, and we have a integrated PhD program where you have a master's degree as well as a PhD degree, combined degree, and we have a very active PhD program. So currently in RCB, it's a research center, so we have around 25 research faculties and around 120 students, which include PhD students as well as postdocs and the project staff. So with that, I will with that introduction, and this is the we have the plant. There are 5 faculties with the background of plant biotechnology. There are 4 chemists. There are 4 structural biologists, and the development biologists, cell biologists. So we are covering basically different areas of basic biotechnology.
Okay. Great.
So with that, I would like now to invite Dr. Rode to formally introduce Professor Disney. Rode.
Thank you, Dr. Bajaj. So it is my great honor and pleasure to introduce Professor Matthew Disney. So Professor Matthew is an exceptional chemist and translational scientist working at a Department of Chemistry, Scripps Research Institute. So let me tell you a little about Professor Matthew's background. So he earned his BSc in chemistry from University of Maryland. He did PhD at the University of Rochester and postdoctoral studies at the Swiss Federal Institute of Technology in Zurich. He is basically trained as an RNA biophysical chemist. So as we know that most of the drugs in the market today focuses on targeting proteins, but Professor Matthew Group invented novel methods that focuses on targeting RNAs to treat many undruggable diseases. So targeting RNA has a great therapeutic potential. So in this research area, he has authored around 159 scientific publications over 8,000 citations. So it is so impossible to mention his every single accomplishment because they are a lot. Some of them I am mentioning. So he was awarded a prestigious International Sackler Prize in 2019 for his outstanding contribution in physical sciences. He has received numerous other awards, including NIH Director's Pioneer Award in 2015, Weaver H. Gaines Entrepreneur of the Year Award in 2018, Scripps Florida Outstanding Mentor Award in 2017, Tetrahedron Young Investigator Award in Bioorganic and Medicinal Chemistry in 2016, David W. Robertson Award in Medicinal Chemistry in 2014, Eli Lilly Award in Biological Chemistry in 2013. So in addition, he also has various editors' responsibilities, particularly as an editor of Bioorganic and Medicinal Chemistry Letters, and also associate editor of Journal of American Chemical Society. I'm sure that we will all enjoy getting to know about his research. Let's give a warm welcome to Professor Matthew. Professor Matthew, I won't take any more of your time. It's all yours.
Well, thank you for the very kind introduction, and thank you all for listening to me on Friday afternoon. So thank you very much. So today I'm going to tell you about work in my lab on drugging RNA with small molecules. I want to start, and this is where I live. So this is like 5 minutes away from Scripps. We live on the Atlantic Ocean. So that was yesterday's sunrise. I think it's pretty amazing to be able to do science and to work with young people. And so this is a picture of my group about a year ago. I'm going to discuss work initially by Peiwan Zhang, who helped us develop methods to drug undruggable proteins by binding to their RNA. And then later on, I'm going to talk about work by Matt Costalis and Haru Akawa on developing approaches to degrade RNA with small molecules by using nuclease recruitment. And then I'm going to end the talk, if I have time, on new work from Ali Angivello and Raphael Benhamou on basically figuring out how to affect RNA processing to trigger degradation of many targets. And I think I want to thank the U.S. taxpayer for enabling us to do science. And so I think it's pretty remarkable that you can write an NIH grant and at least in the US, you can get funded to do the science that you have an idea in your head about and that your peers think is worth pursuing, and be able to, obviously have resourcing to determine if your ideas are good or probably need some adjustment. Okay, I broke this talk into 3 sections for the students, and I have references to the papers that can help them follow if they want additional reading. And so the first module of, I guess, 4 modules is going to be the introductory module, and that's highlighted by this review in JAPS. And so I think, we're living in an amazing time in science. I mean, I think most people would say scientific progress is very slow, but I don't think that's correct. I mean, there's no year in the history of humankind that has showed us that science can progress things at, to use a US quote, warp speed. And so, there was an pneumonia that was discovered and told to the world in Wuhan, China by the World Health Organization on December 31st of 2019. The genome of that virus was sequenced and deposited on the United States PubMed open access portal on January 12th. The next week, people had analyzed and were starting to determine how to make vaccines against us. And in the next month, there was a candidate vaccine by both Pfizer and Moderna. And by the end of the year, most amazingly, this was approved to be put into man. We would all like this to be faster for obvious reasons, but I think we need to really recognize that there's a few constants in our life. One of those constants are we're always going to be dealing with challenges of significant importance, not only in our personal lives, right, but also as a global community, which we most certainly are now. We weren't in the past, but we certainly are now. And the only way that we can address a lot of these issues, I think, in a robust and honest and fair way is through science. And science keeps getting better. It's better today than it was yesterday. It's going to get better tomorrow than it was any other time in previous history of man on Earth, and it will continue to do that. One thing that has struck me in terms of the progress of science is comparing the efficiency by which we can do computations, and that computer processing is enhanced. This is Moore's law, and so it's a constant rate. Computational power doubles every year or two, and that's been constant. When we look at, however, the rate at which we've been able to efficiently acquire genome sequencing information, it far outstrips the efficiency in computational power. And so the consequence of this is, about 51 years ago, a man was put on the moon with computational power that's 1/100,000th of what all of us will have in our pockets, our iPhone, or on our wrist in a smartwatch. And I think that's pretty amazing. One of the most reasons in which these efficiencies have occurred is because of, frankly, the impact of chemists and biotechnologists that have figured out ways to quickly acquire the first draft of the human genome and then define next generation sequencing. One of the most important implications of the Human Genome Project, and it's certainly one of its articulations as to why to do it was to understand the association of genes and disease. And one of the most interesting, I think, probably the most interesting, certainly from my perspective, implications of the Human Genome Project was that it was known that only about 1% of our genome encodes for protein, yet about 80% of it is transcribed into RNA. And many of those RNAs play extremely important roles that go beyond simply coding for those 1% of the genome that gets turned into protein. So we know that there are non-coding RNAs, et cetera. We also know that organism complexity correlates not with protein coding content, but it correlates with the number and diversity of non-coding RNAs. And we also know that many diseases are associated with aberrant expression of RNA, and these include very important diseases. COVID has an RNA genome, but also diabetes, cancer, ALS, all the diseases that you can think about that really are causing major issues for humanity can be addressed by a small molecule targeting RNA. And yet, most of all activity in making medicines that are small molecules has been focused on proteins. In fact, most people would say a druggability problem is solely focused on protein, and that's because only about 15% of our protein families have been drugged with a small molecule. There are significant consequences of that. So for example, this morning, about an hour ago, literally, a mother, one of the largest undruggable proteins, there was clinical data that was shown that you could drug it. And a lot of this work was enabled by chemical biologists. So for example, Kevan Shokat at UCSF showed that you could get covalent small molecules that bind to and activate mutant KRAS in a very potent and specific way. And that inspired Mirati, which is a biotech company, and Amgen, which is a huge biotech company, to develop programs to lead optimize those covalent molecules. And this morning, in fact, the efficacy data for that Amgen molecule was published. And it is very clear that the US Food and Drug Administration is going to have some pressure on them to quickly approve this molecule because it could help patients. It could help humans that have cancer, and about 90% of cancers is associated with a mutant KRAS, and this would affect a very large percentage of them. However, if we look at RNA, about 0% of it has been drugged, and yet I just showed you that nearly every disease could be affected with a small molecule that targets an RNA structure. Now, I would be remiss if I didn't mention this, but there's been a lot of activity historically over the past 20 years in drugging RNA, not by using small molecules, but by using antisense oligonucleotides, by using initially a method that was described by Paul Zamanek, a historical figure in the nucleic acids community. Paul and his group discovered transfer RNA. And later by work by Mello and Fire on using siRNAs. But those target sequence, we want to target structure. And so the way that me and my lab view RNA as a druggable entity is very centrally focused on the folds that RNA adopts. So RNA is a single-stranded biopolymer that folds onto itself. The way that we look at an RNA target, and this is generally how all RNAs fold, is they have regions of low probability of structure. That is, those are regions in the RNA that are not folded. And they also have regions of high probability of structure, regions that are very robustly folded. And what our lab over the past 16 years has deployed and delivered are computational approaches that can ask the fundamental question about molecular recognition, namely, is a specific RNA fold druggable with a small molecule? Does it have a druggable pocket in it? And the way that we define that, as I'll show you in the next, say, 5 to 10 slides, is that we can deliver a small molecule that will bind to that druggable pocket by rational and predictable means. And by binding to that druggable pocket, we could affect the biology of an RNA. In addition to computational tools that can deliver high quality chemical probes that bind to an RNA target, we've also developed a series of companion methods that cross-link small molecules to their RNA targets in cells. And we can purify those cross-linked RNA small molecule interactions And those cross-linked interactions can allow us to identify what are the specific RNAs in a cell or an animal that are bound by a small molecule, but they can also identify what are the sites of binding. And that's really important. The other thing that we've been able to discover throughout the course of these investigations, which was not generally known, is that RNA, small molecules can bind RNA very specifically, but they will not necessarily elicit a physiological response. In cases where binding is very avid, but binding is not enough to affect the RNA, we have developed an approach called ribonuclease targeting chimeras, where we can append small molecules that bind to RNA with a secondary small molecule that will recruit and activate cellular ribonucleases to catalytically and substoichiometrically degrade RNA targets. And I'll talk about all of this in this talk. So one of the questions you could ask yourself is, how in the world do you identify by rational means what are the RNAs that fold and are bound by small molecules? Well, the way that we go about doing this is called Inforna. And what Inforna houses is a database of annotated interactions between small molecules and RNA structures. And the way that this works is we've developed an experimental approach that allow us to identify and annotate RNA fold small molecule binding interactions. The way that this works is we can screen libraries of small molecules for binding to libraries of RNA targets to identify pairs between the two. And so what we have is a RNA structure that contains 4 to the 6th or 4,096 combinations, which is shown here to the left. And we label that RNA target with a fluorescent label, and we hybridize it to small molecule libraries in the presence of these unlabeled competitors. And the way that we do that is we take these RNA folds and put it on a small molecule microarray in the presence of these unlabeled competitors. And what this allows us to tell, because small molecule microarrays allow us to spatially distribute small molecules at different positions on a slide, it tells us which small molecules bind to a member of the RNA library. And then what we can do is we can go in and excise the bound RNA targets, and that allows us to run next generation sequencing to identify all of the RNA folds that bind to a given small molecule and annotate them for affinity. The way that we do annotation is we do deep sequencing of the starting RNA fold library, and we identify the relative frequency of each of those motifs in the library. And then we correlate that frequency from the starting library versus the frequency of each motif in the selected library. The RNA folds that are higher enriched in the selected library versus the starting library are higher affinity binders. And by using these interactions, we've been able to show that we can inhibit the binding of proteins in both cells and animals to RNA targets. We can inhibit translation in both cells and animals. We can activate translation in human iPSC stem cells in cells and animals. We can epigenetic modify gene targets in iPSCs and Fragile X syndrome. We can modulate splice sites in both cells and animals, and we can degrade a messenger RNA in cells and animals. Here's an affinity landscape for bosutinib, which is an FDA-approved kinase inhibitors. Known drugs bind RNA. When we have a Z-OBS, which is basically a score for that enrichment in the selected library versus the starting library, this molecule, if the Z-OBS is above 8, it's avid binding between RNA fold small molecules, and this compound binds 18 motifs of 1,024. We then put all of these interactions into Inforna, which I'll show you later, basically to decode which of those motifs are present in biological systems. And we've been able to do that, and bosutinib will bind these 3 RNA targets. By using this approach, we've been able to identify a wide variety of small molecules that bind to RNA. These small molecule RNA interactions include known drugs, novel compounds, and fragments and scaffolds. So we have a broad information base by which to understand not only the RNA folds that bind small molecules for each one of these individual targets, but we can broadly identify what are the types of small molecules that bind to RNA folds. And so we deployed this approach to target an undruggable protein. So one of the questions that we had early on in this program is, could we affect the levels of an undruggable protein by binding to the messenger RNA that encodes for it? So in this sense, we could expand druggability in the proteome by targeting RNAs that encode for these proteins. And the protein that we selected is alpha-synuclein. And so alpha-synuclein is a undruggable protein, and it's undruggable because this protein target does not fold into a structure that's well defined for binding to a small molecule. Furthermore, and that's because it's an intrinsically disordered protein that doesn't have a specific fold. Parkinsonism can be caused by alpha-synuclein overexpression. In fact, it can be triplication of this gene. And then this alpha-synuclein will form deposits in Lewy bodies in Parkinson's disease, dementia, and multi-system atrophy. And there's been a wide variety of approaches that have been put forward to affect alpha-synuclein expression and aggregation. Most of these, however, have targeted aggregation-prone alpha-synuclein in an extracellular matter or have used oligonucleotides that are delivered intrathecally. The way that alpha-synuclein messenger RNA folds, that makes it amenable for small molecule targeting is shown on this slide. And so alpha-synuclein has a regulatory structure in its 5' untranslated region called an iron responsive element. This iron responsive element functions by binding to proteins called the iron regulatory protein, and the interaction of the iron responsive element with iron regulatory protein inhibits translation of alpha-synuclein messenger RNA. So under conditions, however, of high iron concentration, alpha-synuclein will, its translation will be activated because iron will bind to this protein. And that will, so iron will bind to this protein and inhibit, iron will bind to this protein and inhibit its translation. Iron will bind to the protein, free the protein, and then it will become translated. So the question is, could we design a small molecule that would enter these cells and affect translation specifically? And so we utilized Inforna to identify within these regions of structure what RNase sites could be targeted with a small molecule and what are the small molecules to target these RNA sites. We identified 2 regions in this structure that could be bound with a small molecule. I'm going to focus on this yellow region because it turns out this region you can affect selectively alpha-synuclein translation. So for example, this molecule synucleazide binds to this region in the RNA, and it's very unique across the transcriptome, this site. 2 microRNA precursors contain this RNA target, but they're not expressed in the central nervous system where we want to get to affect alpha-synuclein. And 3 transfer RNAs also contain this motif, but they're not expressed either. When we deliver this molecule to glioblastoma cells that express alpha-synuclein, we can inhibit its translation with an IC50 of about 500 nanomolar. And that's very selective in that we only inhibit alpha-synuclein. We don't inhibit the translation of other iron responsive element containing RNAs. And that's because the region within this IRE that we're targeting, namely that A bulge and the 4 base pairs that surround it, is unique to alpha-synuclein's IRE. The molecule does not inhibit alpha-synuclein translation if we mutate or remove the iron responsive element, suggesting and supporting that the molecule affects translation by binding to that target. And last but not least, the compound does not affect the levels of the iron regulatory protein. Because if IRP was upregulated, then you could imagine that that upregulation of that protein as an off-target for this molecule could inhibit translation. What we also know is that this small molecule that inhibits translation has a selectivity profile, at least at the level of RNA-seq, that is competitive with an siRNA that inhibits alpha-synuclein. For example, this siRNA will inhibit alpha-synuclein, that's shown here. Synucleazide, the small molecule that we developed, will not inhibit translation, and its similar number of genes are affected across the transcriptome. That doesn't mean that the small molecule is as selective as functionally selective as the siRNA. It's just to put out there that we can get fairly selective molecules that affect translation and have activities against RNA. The other thing that we've been able to do is that these molecules will trigger cell death when these compounds are treated with preformed fibrils. And so in this experiment, you can treat cells with preformed fibrils of alpha-synuclein, and the endogenously expressed alpha-synuclein will form aggregates with those preformed fibrils and cause cell death. So if we're indeed inhibiting alpha-synuclein translation, not only should we see a reduction via Western blot, but we should see a reduction in the ability of these compounds to trigger apoptosis in cells. And that indeed is what we see. Apoptosis is measured by LDH release. And when we deliver these molecules to SHSY5Y cells, we can see that the LDH release is greatly diminished within these cells. So we can not only inhibit alpha-synuclein's translation, we can also very specifically affect an alpha-synuclein associated phenotype, namely LDH release. We went to further investigate what was the mode of action of this molecule by doing something called polysome profiling. And what polysome profiling is you can basically take cells that are treated or not treated with drugs, and you can separate based on sucrose density gradients, the messenger RNAs that have different densities. So that is messenger RNAs that have higher densities of ribosomes will have a different mobility within the sucrose gradient. And what you can do is run qPCR throughout that gradient to identify where the messenger RNA is. And by doing that, you can understand what is the effect of the small molecule on alpha-synuclein messenger RNA in terms of ribosomal density. And so there are 3 steps in this that I want to point out with you that are modulated with our small molecule. One is ribosomal translation occurs by having a pre-initiation complex where ribosomes will assemble on the 5' end of a message. They then read through structure. Here's the IRE in alpha-synuclein's message, and the start codon is here. Once these pre-initiation complexes read through the start codon, they'll assemble into active ribosomes that begin translating. And then this cycle is repeated where monosomes that are on ribosomes get converted to polysomes. And by doing a polysome gradient expression with and without, polysome gradient experiment with and without drug, we've been able to show that we've stimulated the amount of alpha-synuclein messenger RNA that is in the fraction with incomplete ribosomes. And also we've diminished the amount of polysomes that are associated with alpha-synuclein message. So basically the mechanism of this molecule is thermodynamic stabilization of that iron responsive element messenger RNA and inhibition of read-through translation of that messenger RNA. What we've also done is we've looked at to understand how broad this potential mode of this activity could be across the human transcriptome. And so what we did is we looked at all messenger RNAs that are encoding an intrinsically disordered protein. And so you can go and look at, and there are servers that have identified all messenger RNAs that have identified all intrinsically disordered proteins. And so we can look at all of those intrinsically disordered proteins and then look at the messenger RNA that encodes them and ask a fundamental question, which is, do the messenger RNAs that are encoding intrinsically disordered protein have some structure within them? And it turns out that about 95% of them do. Now, that doesn't mean that binding to that site will affect the translation of the messenger RNA, but it does suggest that this approach could be broadly applicable to other important diseases. So that's important. And so the summary here for this section of the talk is that targeting, we can target messenger RNAs that encode for undruggable proteins with small molecules. And we can do this in very, we think selective and important ways that can expand druggability. In the case here is that we're able to target and inhibit translation by targeting an event that's near a start codon. And therefore it suggests that inhibition of translation could be affected by targeting messenger RNAs near a start codon. And there's a lot of work to be done on this compound. It's just a tool molecule, and we have to enhance it for druggability. The other question that we wanted to address within our program is, could we figure out methods to degrade messenger RNAs by subjecting them to quality control? So our messenger RNAs are basically built to be destroyed. And the manner in which they're built to be destroyed is by basically, there's a ton of ribonucleases that our cells express to degrade RNA. And so the question we had within this paper, which is highlighted here, is could we unnaturally force messenger RNAs to be recognized by RNA quality control? And this is important to target RNA because many RNAs are associated with important diseases, including cancer metastasis. And so our major question was, could we utilize this technology to destroy an oncogenic non-coding microRNA that causes cancer metastasis? And the non-coding microRNA that we chose to pursue in this endeavor is microRNA 21. MicroRNA 21 is a non-coding microRNA that is upregulated and expressed in triple-negative breast cancer. What happens for all microRNAs is that they're produced as primary microRNAs, where they have what's called a drosha site, and the site nuclear nuclease drosha will bind to that site and cleave the prime microRNA. That will liberate a precursor microRNA, which gets translocated to the cytoplasm, which is cleaved by the cytoplasmic nuclease dicer to liberate a mature microRNA. That mature microRNA will get loaded into Argonaute protein, which will silence messenger RNAs that have 3' untranslated regions that can form Watson-Crick-Franklin base pairing with this 3' UTR to silence translation. One way in which you can inhibit microRNA biogenesis is by binding to the dicer or drosha sites, which we've been able to do here. And by binding to those sites, you can inhibit the recognition and catalytic activity of dicer and drosha onto these targets. And the end result is you'll increase the levels of a protein rather than decrease. We then looked at to identify a small molecule that could bind to microRNA 21 by using Introna, is we analyzed its RNA structure by using our tools. The dicer site in microRNA 21 contains 20 microRNA precursors that contain this motif. The adjacent site has 39 microRNA precursors that contain this motif. However, one microRNA precursor contains both of these motifs. And so if we could identify a small molecule that would bind both of these motifs, we could link it together and have potentially a selective ligand that targeted microRNA 21. That indeed is what we did by using Introna. So we identified not so low molecular weight fragments that bind to this RNA target. We custom assembled them and then deployed them for affecting microRNA 21's activity. This is data in triple negative breast cancer cells for the expression of all microRNAs in these cells. In orange is the activity for the monomer, which you can, in this dotted line is a false discovery rate of 1%. What you can see is that the monomer is not as selective as the blue dot or blue purple dot, which is the dimer. Furthermore, this is a similar inhibitory activity, and the dimer is about 10-fold more bioactive than the monomer is. As I mentioned earlier, we wanted to see if we could utilize this technology to interface unnaturally RNA targets with quality control machinery. And the way in which we went about doing that is something called ribonuclease targeting chimeras, or RIBOTACs. In this approach, what we did is we linked a secondary small molecule onto this RNA target. And that secondary small molecule would basically bind to and activate RNase L that would cause catalytic and substoichiometric degradation of RNA targets. A small molecule recruiter of RNase L would work in the following manner, is that it would bind to RNase L, and that binding to RNase L would activate it and trigger its degradation. On the top here is the dimer structure for binding miR-21. On the bottom are these active and inactive RNA cell recruiters. I should call this less active. If we go into triple negative cancer cells, the triple negative breast cancer cells, what you can see is that in gray is the activity of our dimer, in orange is the activity of the control degrader, and in green is the activity of the active degrader. The active degrader, all three of these compounds inhibit the levels of microRNA-21, but the gray and the orange compounds should not facilitate degradation. So in contrast, if we look at the precursor to microRNA-21, these levels are increased. And the increase in those levels is because we're inhibiting the processing of this target. However, if we look at the degrader diminishes the levels of this target, which is supporting that the compound is degrading the RNA. We've also completed in this paper a wide variety of control experiments to validate that RNase L is indeed the nuclease. So one is we've taken CRISPR or siRNA knockout of these cell lines and shown that when we do that, we ablate and inhibit the ability of the compound to degrade the target. The other thing that we've been able to do is we've immunoprecipitated or taken an antibody to RNase L. And when we add the compound to cells, we're able to immunoprecipitate the precursor to microRNA-21. So all of this suggests that RNase L is our active ribonuclease. One of the consequences of degraders, which frankly surprised me, is that you could imagine a situation where when you start to invoke degradation, you would actually lose selectivity rather than gain it. And what we've been able to observe is that when we facilitate degradation, we actually enhance the activity of the molecules. These are similar inhibition levels in, again, orange is the monomer, purple is the dimer, green is the degrader. You can see that there's similar activity, but the concentrations are dramatically different. In fact, when you go from a binder to a degrader, that degrader is about 20-fold more biologically active than the binder, and it's 200-fold more biologically active than the monomer from which it was derived. Furthermore, as you can see from this volcano plot, is that the selectivity is actually enhanced when you have this degrading molecule, and that selectivity is represented by what's called a Gini coefficient. The other thing that we've been able to do is look at in cells, what is the effect on the proteome and the transcriptome when we deliver these small molecules? This is one thing that's really important to do in these experiments, is you really have to understand what is the effect on the proteome and the transcriptome in profiling experiments. And so what we've been able to show, and this is the, when we deliver the degrader at 50 nanomolar concentrations, we can see that a large number of the anti-proliferative proteins are upregulated in their expression, and a large number of the proliferative proteins are downregulated. We basically changed a complete oncogenic circuit by inhibiting the endogenous levels of microRNA-21. Furthermore, if we go and look at the levels of every single protein in this proteome and study if they have a microRNA-21 binding site, if these proteins have a microRNA-21 binding site, the hypothesis is that if you inhibit microRNA-21, they should be upregulated in their expression. And that is indeed the case. That's what this green dot is that that's the level of all proteins that have a microRNA-21 binding site are upregulated, whereas in contrast, proteins that do not have a microRNA-21 binding site are not affected. Furthermore, in the last two slides, we conducted an animal study. And in this animal study, we delivered triple negative breast cancer cells to the tail vein of a mouse model. And what happens is, if you do that, eventually the breast cancer will metastasize to the lung. And that metastasis is shown here. Each one of these dots that I have highlighted with this red laser pointer are cancer metastases that occurs to the lung. When we deliver IP, 5 mg per kg, this degrader, we diminish the metastasis in that lung greatly. In fact, here's data for 9 mice. Is the number of nodules has greatly diminished, but this is, I think, under representing the activity of the molecule, because we're just scoring if a module is there, not necessarily its size. Furthermore, we did histology staining, where we took a lung and sliced it into pieces and then stained. And you can see that the HE staining within these mice is dramatically diminished. This is cancer, that staining. And last but not least, we wanted to see if there was a pharmacodynamic or an effect directly on the target that we could measure in histological samples retrieved from these mouse models. And so when we do that experiment, we can see that microRNA 21 is diminished by fluorescence in situ hybridization. Remember, the degrader is degrading the precursor to microRNA 21, and that precursor is also diminished by fluorescence in situ hybridization. And if you'll recall, microRNA 21 silences PDCD4 protein, which is a pro-apoptotic and anti-invasive protein. And you can see that that protein is upregulated also in this histological sample. And last but not least, and I should have mentioned it at the beginning, is that these degraders are well tolerated in these mice. There's no issue whatsoever with them. And so I want to tell you, this is the tip of the iceberg for where we want to go with this technology. And apparently it looks like we can identify chimeric molecules that can interface with native quality control to affect biology quite broadly. The last thing that I want to end here, and I'm not going to go into this, I'm going to go through this very quickly so that we have time for questions, is one of the scientific oddities that I challenged my lab to pursue early on was, could we convert binding molecules into degraders? That is not necessarily directly recruiting and activating the ribonuclease activity, which in these cells, but rather could we affect RNA processing to directly facilitate the degradation and elimination of toxic RNAs. And so we wanted to apply this technology for a class of, I think, really important and very challenging biomedical issues that the community has largely ignored. And that is targeting RNA repeat expansions. And so RNA repeat expansions cause greater than 40 different genetically defined and rare diseases. These include things like Huntington's disease, C9orf associated ALS, and also myotonic dystrophy type 1, the most common cause of adult onset muscular dystrophy. And the CUG repeat that causes myotonic dystrophy type 1 also causes the front of the eye disease, where you have to get a corneal transplant. And so we were able to design molecules that bind to these targets in CUG. And so the question we had is, would a CUG repeat expansion binding molecule in the 3' UTR of an untranslated region, would that compound also be applicable in Fuchs' corneal dystrophy? Now, the reason that RNA repeat expansions, namely CUG repeat expansions, cause disease is because these repeats sequester proteins that are involved in RNA splicing. And long story short, this is natural splicing. The protein that gets sequestered by the repeats bind to a pre-messenger RNA, you get natural splicing. When you don't have proteins on this pre-messenger RNA, you get a defect in that splicing that includes a stop codon, and that messenger goes away. If we can deliver a small molecule that would kick off the binding of muscleblind onto these proteins, you could get natural splicing. And so we delivered and studied the structure of a small molecule that binds to this target. That small molecule, when we deliver it into both myotonic dystrophy type 1 cells and Fuchs' corneal dystrophy, will improve the defect in RNA splicing that's due to muscleblind sequestration. The reason that I'm presenting this is, what's interesting is that in myotonic dystrophy type 1, it's a 3' untranslated region that causes disease. However, in Fuchs' corneal dystrophy, it's an intronic RNA that causes a disease. And what you can see is that the level of this DMPK message is unaffected with the binding small molecule, but the level of TCF4 message is greatly depleted. And that is because it turns out that in FECD, the reason that this intronic RNA causes a disease is because the intron is harbored as a retained intron, and it is not subjected to the normal decay pathways that introns have. And so what we know is that the small molecule will bind to this intronic RNA, and it will liberate it from the pre-message and shunt it to the RNA exosome to facilitate its targeted degradation. And so basically, I'm going to end there, but long story short, I think there's a lot of ways to affect RNA with small molecules. And these compounds not only allow us to understand what are the biological roles of RNA, but I think for some very important diseases, they allow us to put forward methods that can help us to articulate targeted therapeutic options. And so I want to also end by thanking my group again. Again, Peiwan Zhang spearheaded the work on inhibiting, affecting druggability by targeting messenger RNA. Metastasis and Haru. Haru's going to go for an assistant professorship in University of Tokyo in the next few months. Matt works at Dicerna. We got collaboration from Hafiz Hanif, who's at Skyhawk Therapeutics. Ali Angibello is at Expansion Therapeutics. Simon Benhamu is at, he's going to start a faculty position at the University of Jerusalem. Andre is at the University of Bonn. So thank you for taking the time. I'm happy to answer questions. I realize there are some already in the chat, and I don't know how you want me to go about answering those questions, but I'm happy to do whatever. All right, let me see if I can hit escape. Okay. All right.
Thank you very much, Professor Matthew,
Yep.
for this amazing talk. So we have a couple of questions that have been submitted by the audience. And
Yep.
as a reminder to audience, if you have a question that you have not submitted yet, please enter it in the QA chat box, and we will get through as many questions as time allows. So Professor Matthew, the first question for you is, currently several RNA molecules as a drugs such as siRNA or aptamers are in clinical trials. How do you see the difference between the small molecule versus RNA-based drugs in terms of success rate and affordability?
Okay, I have no idea about success rate. Oh well, okay, let me, you're gonna have to answer these questions because I'm trying to get into the chat, but Zoom for me crashed, so I hope I'm here for the duration.
Yeah.
So, yeah, small molecules will enter tissues that siRNAs won't. So I can tell you, Biogen, Ionis, and Alnylam are working on generally targeting diseases of the liver and kidney, which are highly accessible to antisense oligonucleotides, or the CNS when they directly inject these into the spinal column. And so I can tell you things like cancer or things like muscle disease are generally not, muscle and cancers are generally not exposed to antisense oligonucleotides. They're generally more accessible to small molecules. And so I think one of the ways to articulate this is that there's going to be tissues that tissues and therefore diseases that the small molecules are going to be more amenable to than the siRNAs. And that goes beyond dramatically the convenience. siRNAs are going to be injectables. Small molecules are going to be pills. It is much more preferable for a human to swallow a pill than to go to get an injectable because they just won't get their medicine sometimes. And it's especially debilitating when you get that injected into the spinal column. Now, you also asked the questions about cost. So I can tell you delivery of goods is that the price of making a small molecule, these small molecules, is a lot less than making an antisense oligonucleotide. And furthermore, the formulation is much more simplified. Although I'm projecting a lot, there is high potential for the small molecules to most likely have a lower cost of goods and have a higher potential to be more affordable than an ASO. And I think I answered all your questions.
I hope so. The second question is, RNA function is often linked to its conformational dynamics rather than to unique structures. So how to target a specific structures in that conformational ensemble?
Yeah, so our molecules target specific dynamics in an ensemble and they lock them out. But the reality is, for example, I guess the most work on conformational ensembles has been by Hashimi on HIV TAR. And I can tell you, even though ligands will lock out a specific ensemble, if you bind to that RNA target avidly, it's done. Like you're going to inhibit the activity of HIV TAR pretty broadly.
Yeah.
So, yep.
Yeah. The next question is, in comparison to protein as a drug targets, which consist of total around 22 amino acids. So RNA just consists only 4 nucleotides as a building blocks. Thus the 4 monomeric nucleotides seems less variable for the specificity of drugs.
Great question. So yeah, that's I think that's one of the challenges, but there's other, so RNA will robustly fold, and I can tell you that some of these folds are very unique. The other thing that makes certain RNAs more druggable than others is their expression. If they're higher expressed, it's easier to occupy. So even though another RNA target could have a similar fold, you're gonna more occupy and therefore more inhibit the higher expressed one. I think this argument that you're making, which is an extremely important one, is not so different than an early argument that was made in the kinase inhibition space, is that kinases generally have an ATP binding pocket and a protein binding site. And so you need to be aware of selectivity within them. You know, I think there's more work to be done, obviously, on RNA targeted small molecule medicines, but I do think the fact that we have ChemClip and these other tools can allow us to really understand what RNA targets are being occupied by small molecules versus others to try to further enhance the selectivity of the compounds. So anyway, we'll see. That's a comment that I think the only way you're going to address it is by specifically targeting an RNA with a small molecule. And I would put forward some of the selectivity data that I showed you is pretty compelling, but others may think differently, and that's fine.
Yeah. The next question is how the epigenetic modulation can be regulated using small molecules.
What was the beginning of that question? I'm sorry. How would be a representative?
How the epigenetic modulation can be regulated? Yeah.
Okay, so this is a paper. It's a collaboration with Sami Jaffrey and I that came out in 2014. So it turned Fragile X syndrome is the most common cause of genetically defined autism that we know of to date. And it's caused because you have a large repeat of CGG that's in the 5' UTR of FMRP encoding messenger RNA. And in these patients that have Fragile X syndrome, they don't have FMRP protein. And that's because before, in a pre-differentiated state, that is if you have stem cells that will go and be differentiated into neurons, during the course of that differentiation, that FMRP messenger RNA is produced, and it has this repeat. And this repeat actually binds to the promoter that translates, or the promoter that facilitates transcription of that gene, and you wind up getting an RNA-induced epigenetic silencing. If you take small molecules that bind to this RNA before they're differentiated, you will inhibit the production of those epigenetic silencing marks, and that gene will continue to be turned on. If you apply an RNA-targeted small molecule and a low dose of an epigenetic modifying agent, you will erase and inhibit having those epigenetic marks in the genes and will reactivate it. Yep, so that's how that works. I know that that was a mouthful, but okay.
The next question is how the bioavailability profiles of these repeat degraders in mice, would the effect of degrade vary between different cell types in the tumor microenvironment? Yeah.
Great question. It does. So that's exciting. I think that's exciting. That's not a limitation in my opinion. So you can get, depending on the expression of the target and the expression of RNase L in these cells, you can get very different, very, very different activities. In fact, you know, the sensitivity of these compounds and selectivity goes way up if a certain cell line expresses RNA cells. So we know quite dramatically what our specific cell lines that express RNA cell, and then this could be another level of precision medicine to deploy against them. So yes, that is an effect, and that's an important one to understand. Yep.
The next question is again regarding the degraders. So whether these degraders can be used to stabilize one of the splice variant during alternative splicing, how you will confirm the specificity of targeting alternative splicing, or can it be used to stabilize one of the splice variants during alternative splicing?
Yeah, we've done that. Yeah. So we've done it in Tau. There's a JAX paper on that recently that's published. We've also done it we've made that into a very important molecule that works in a mouse model. Mm-hmm. way that you can study selectivity. That is not a degrader, it's a binder because it's affecting a splicing outcome. The one thing within that we can study selectivity is you basically do RNA sequencing analysis and study the protein splice isoforms with and without the drug. That's pretty straightforward. Yep.
Okay.
We've done a lot of that.
All right. The next question is, does same nucleotide inhibit the scanning process before recognition of start codon, or it inhibit binding of 40S to 60S ribosomal after initiation codon recognition?
Oh yeah, that's a great question. We don't know a lot of that. We think it inhibits scanning. The current hypothesis, although there's more to be done, I was surprised actually that there was not a lot of work done on this, but we think it inhibits scanning through and assembling on the active start codon. We have more work to do to really nail that. That's a great question. I was pretty surprised, frankly, that I didn't think these compounds would be useful tools to study ribosomal scanning, but apparently they are. Yeah, we don't know, and we need to be thinking about that. Yep.
Yeah. The next question is, there are a lot of RNA structures throughout the transcriptome. Then how do you maintain specificity of a small molecule to druggable pocket?
Yeah, so it depends. Some druggable pockets are common, some are unique. We go after the unique pockets. The other thing is that not all targets we're going to inhibit their activity. Binding isn't necessarily enough. And so even if you have an RNA structure that has the same fold in it, that doesn't mean its function is going to be affected. The other level of selectivity that we've been able to impart in these is that when we go to use a degrader, is that the enzymes have a specific molecular recognition structure. And that specific molecular recognition structure that they bind to can further enhance selectivity of these molecules. That is, not all RNAs are going to be degraded. It has to have a specific structure that's a certain distance from the binding site. Yep.
The last question. While targeting RNA sequence with small molecules, will they also interact with DNA given their similarity with nucleic bases and phosphoester backbone?
Some of them do, but we eliminate those. So one in our pipeline is we eliminate molecules that bind to DNA and put forward compounds that just bind to RNA generally. Although, to be frank with you, there are some instances where it wouldn't matter because DNA is in a nucleus and RNA is nuclear and cytoplasmic. And so if you had a ligand that didn't get in the nucleus, you would still be a selective and useful RNA-targeted molecule, assuming it would hit a cytoplasmic RNA. But yeah, we think about that all the time. Yep.
Yeah, I hope I have read all the questions came from the audience. So if there are no more questions, so I would like to thank you, Professor Matthew, on behalf of RCB Organizing Committee, for making time from your busy schedule and gracing our webinar series with your excellent talk and sharing with us your important findings. So I would also like to extend my thanks to student and faculties and our IT support team to be with us at this odd time. So around 60 people are attending this webinar now. So thank you very much, Professor Matthew. So here we'll close today's session. Thank you very much, everyone.
Well, thank you all and have a good weekend and good night. And I appreciate all the very smart questions. So thank you.
Yeah, thank you. Thank you.
See ya.
Yeah.
Thank you.
Yeah.
Bye. Huh.